And the immune system has many components, but probably the most important are leukocytes. This is a type of cell that is constantly patrolling our body and in the search for infection, searching for pathogens. So the capacity of leukocytes to migrate is crucial. My PhD project started with the development of a microfluidic tool to study chemotaxis of these cells. After developing this new tool, now we are starting to apply it for studying the migration towards different signals. Chemotaxis is a very important process where cells sense certain molecules that are diffusing from a given place, and therefore they make a gradient. So there is a higher concentration on the target's place, and the cells would follow. We developed a microfluidic device. This means that it's a very small chip, a few centimeters large, where there are micron-sized channels. And in these channels we can load our cells and therefore put a single stimulus at a time, for instance, make a gradient, and at the same time record the migration of the cells responding to this gradient. So in order to build this tool, we pour the liquid resin and then it becomes solid. And then what we do is that we need to clean the surface and also prepare a glass slide, which is also cleaned. And we activate the surface with a plasma oven. And by doing so, at the molecular level, the surfaces are active. And then when we put them together, they will glue. And this is a molecular bonding, so the device is sealed. And once we have our devices, we can coat the substrates the way we want to test. And at the end, we put the purified cells from blood. So we take blood from healthy human donors and put the whole device under the microscope and record cell migration. Alternatively, we want to investigate how cells respond to signals which are anchored on the substrate. And to this end, we use the tool that is commercialized by our industrial partner, Alveol. And this is a technique which is based on photo patterning. And therefore, we can design any kind of gradient or shape of interest. And then by illuminating with UV, we can activate the substrate only on those specific areas. So I studied biotechnology in Argentina. And this is a career that is already quite diverse because we study from molecular biology to immunology and quite some physics as well. And right now my PhD is in biophysics, so I try to move more into this direction.